I really do have sort of two jobs, one as a university professor and one as the chief science officer for a company. And as I give my talk today, I'm going to draw on examples from both entities, how we're developing the technology, advancing it, trying to engineer plant genomes with higher efficiency, and then how we're applying it sort of in the real world to address problems of agriculture. And with respect to agriculture, we feel a particular burden to develop techniques and technologies that are really going to address the increasing need for food, particularly in light of the world's expanding population. So as we approach mid-century, we're going to have 9 to 9.5 billion people on the planet, and this is going to tax agriculture and plant agriculture in particular. And the demand for food is also going to occur in regions of the world that are already have troubles with food security in terms of reaching the expanding populations in Asia and Africa. And I'm an optimist, and plant agriculture has really been very good at keeping up with the demand, the increasing demand for food. This slide shows increases in productivity in maize, rice, wheat, and soybean, some of the world's major commodity crops. And what we see is this through traditional genetics and more recently through biotechnology, we see an increasing improvement in crop yields. But as we project population growth on agricultural productivity, we anticipate, again, increasing on sort of the same rate, but the demand, the dotted lines here represent where we need to go in terms of production. And so there's going to be increasing challenges on agriculture. And there are many solutions to this problem. One is, increasing really the rate of crop improvement, improved plant varieties that are going to produce better on marginal lands or with fewer inputs, less water, fewer amounts of fertilizer. And so, genetic variability can produce, or deploying new mechanisms to create genetic variability can create crop varieties that can really sort of meet this increasing demand in the food supply. And so we're really applying genome engineering to solve two problems. One, we have to basically understand how plant genes and genomes work, and then take that information and apply it to create genetic variation that's going to be relevant to agriculture, that's going to give us these crop varieties that are going to meet the demands, increasing demands for food produced by plants. So this slide is one that's very familiar to you, and you've been thinking about it all week. That is, your CRISPR reagents create the double-strand break, and we try to direct those outcomes to produce genetic variation of interest. In plants, we have this pathway down really pretty well. Making the break, and then through imprecise non-homologous end joining, we create a mutation that ends up knocking out a gene function. The way we typically mutagenize plants is to create a transgenic intermediate. So this represents the genome of a plant we want to modify, and we insert at some random site in that genome a nuclease, a CRISPR-Cas reagent, for example. And we deliver these reagents by a variety of means. But as those transfected or transformed, as we refer to them in plants, cells grow and divide, that nuclease is expressed. It knocks out our gene. And we take these somatic cells, and plant cells are really inherently stem cells. We can take a somatic plant cell that's been transformed, mutated, and grow up, and through hormone treatments and tissue culture manipulations, grow up an entire plant. And then that plant then has a modification of interest. And then we can segregate away that nuclease, and we obtain just a plant that has our mutation of interest with no foreign DNA resident in the genome. So my title suggested genome-edited crops are going to enter the dining room. And I want to give you a few examples of some of the first types of genome-edited crops that are going to enter the food supply. And these examples are those that are taken from the company that I consult for, Calyxt. And one of the commodities, or one of the plants that we're interested in modifying at Calyxt is soybean. Of course, soybean is a really important food crop, both for livestock and for humans. In terms of animals, it's the #1 source of protein. And so after the soybean seeds are harvested, they're pressed, and a soybean meal is produced that is used as a source of animal feed. But then the oil that's also results from pressing and crushing the soybean seed is used for a variety of purposes, particularly in cooking. And so it's the #2 source of oil for human consumption. Now in the United States, over 95% of the soybean crop is GM. And when I use the term GM, I'm talking about what I would say is a conventional use of that term. That is, it has a GM plant, in my definition, has foreign DNA that's been inserted into its genome, foreign DNA that originates from another organism, right? So 95% of the US soybean crop has DNA from a bacteria, for example, that confers resistance to an herbicide, or genes that confer tolerance to pests and pathogens. 5% of the market is non-GM, and at our company, of course, we're trying to find a niche where we can find markets for our products, and we're focusing on that non-GM fraction of the market. And a problem in soybean is that the oil that's produced is actually not ideal for cooking purposes. It's very low in these monounsaturated fats and very high in polyunsaturated fats, so 23% monounsaturated fats. So as such, the soybean industry, what they do is they chemically treat the soybean oil so that they convert the polyunsaturated fats into monounsaturated fats, so that they create an oil composition that's more similar to canola, olive, or sunflower oil. These oils are very suitable, are healthy, and are very suitable for cooking and frying purposes. But when you chemically treat soybean oil to increase the fraction of monounsaturated fats, a byproduct of this hydrogenation process is the production of trans fatty acids. And these are unhealthy, and the Food and Drug Administration has said that by 2018 there can be no trans fats in the human diet in the United States. And so for oils like soybean, this ruling and the anticipation of this ruling has resulted, starting over a decade ago, in a decrease in the demand for soybean oil. What it's been replaced by are those oils that have a higher composition of monounsaturated fats, the sunflower oils and the canola oils. And so what we at Calyxt tried to do is to try to come up with a genetic strategy to change the fatty acid composition of the soybean oil and genetically create an oil that's more like canola or olive oil. So soybean encodes these fatty acid desaturase enzymes. They take a monounsaturated fat like oleic acid and add another double bond to create linoleic acid. And this linoleic acid can get further modified into other polyunsaturated fats. So there are 4 genes. So soybean is, like many plants, has a complex genome, and 2 of them are expressed in the seed. So those were the ones we wanted to target. And so the idea was, if we knocked out these seed-expressed genes, we could increase the production of oleic acid and decrease the amount of linoleic acid. And our approach was the one similar to the one I outlined a little bit earlier. We would make our nuclease and we would transform it, introduce it into soybean cells, and then we can grow these cells in a petri dish and recover plants, and hope that that the expression of the nuclease during the development of the plant had knocked out our genes, and then we could segregate away the nuclease transgene and create a variety that we would then introduce out into the field. And in fact, we were able to do this with the efficiencies, the types of NHEG efficiencies, very similar to the efficiencies that have been described in human and mouse and zebrafish cells. over 10% and often over 50% of the cells have a mutation in one or more alleles. And so this is now the fatty acid composition of our modified soybean oil. It has those knockouts in the 2 genes, all 4 alleles of the seed-expressed fatty acid desaturase. And what you can see is now the seed oil composition is very similar to our target oils of sunflower and olive. Low in polyunsaturated fats and high in these monounsaturated fats. These particular knockout mutations were made by TALENs, although CRISPR-Cas works at a very similar efficiency. And I just show this slide to remind you of the types of modifications that are introduced through non-homologous end joining, small deletions at the break site. But for a food crop that's going to be introduced into the field and into the food supply, this is a new way of doing business, right? The GM plants that I'm currently talking about all have foreign DNA inserted into them, and now we have applied genetic engineering and biotechnology to create a plant that really only has a handful of nucleotides removed from the target site. And so I've gone before the USDA, I've gone through in front of ministries of agriculture in China and Japan. We've approached Argentina to say, how are you going to regulate a product that's made by this technology? And for the USDA, they've been very consistent. That is, our soybean, the product I just described, a number of other products created with TALENs, meganucleases, zinc finger nucleases, and most recently CRISPR-Cas. All of these products are considered to fall outside of their regulatory authority, and that is the rules and regulations that were put in place to regulate products of biotechnology really don't apply to these products, and they're treating them as if they were mutations made by other means, by EMS, chemical mutagenesis, or X-rays or gamma rays. And so what that means is we can go from the laboratory into the field, and this applies to companies or as well as individuals at universities. We can now create modified plants and get them into the field and test how they behave. And this is in fact what we've done with our soybean, both in the United States, and then we received regulatory approval in Argentina. So we can grow them in the summer in Minnesota, and then in our winter in Argentina. And this is our field in Argentina that was harvested this past spring. And we shipped 30 tons of soybeans that we grew in Argentina to the US and are growing them out now in over 500 acres in the United States. So in 2018, we hope to take our soybean and introduce it into the market, make non-GM soybean oil and meal that's high in oleic acid, that doesn't have any trans fatty acids because you don't have to chemically hydrogenate it. And we believe this will probably be really the first product in plant agriculture that's introduced into the food supply. So I want to give another example in potato. Again, the application of end joining, non-homologous end joining to improve what is actually a very important commodity crop in the United States, amounting to $30 or so billion, principally used for French fries and potato chips, or sold as, potatoes are sold, of course, in the grocery stores as well. So potato is clonally propagated. You don't plant potato seed, you plant slices of a potato tuber. And so for centuries, we've been planting basically the same potatoes. There's been no or very limited genetic improvement. Russet Burbank, one of the most commonly cultivated potato varieties, was developed a century ago. And so the idea here is that you can take this technology and now to a plant that's basically been clonally propagated, use this technology to really add some value to solve some of the problems in potatoes. Storability, about 15% of it's wasted because the potatoes can't be stored very well, and I'll talk about that more extensively. Or reducing browning through the processing of potato to create fries and chips, for example. So the storability problem is the one that we attacked initially. And what happens is when you harvest potatoes, tons of potatoes in Idaho every fall, you can't process them all into fries and chips simultaneously. And you can't store them at a reduced temperature. If you refrigerate a potato, you activate the expression of an invertase, which breaks down sucrose in the tuber into glucose and fructose. This is why when you buy your potatoes at the grocery store, they're at room temperature at the store, and you take them home and you don't refrigerate them. If you do, you start to break down the sugars and the potatoes soften. This is called cold-induced sweetening. And potatoes that are stored in the cold, these reducing sugars, glucose and fructose, they lead to caramelization when you cook the potatoes, and at high temperatures, they react with free amino acids and make acrylamide. Of course, acrylamide is a neurotoxin and a potential carcinogen. And the presence of acrylamide in fried foods is starting to be regulated. Here in California, for example, you're only allowed 300 parts per billion of acrylamide in your food. And many potato chips and French fries processed from common cultivars of potato often exceed the amount of acrylamide that's in California has been deemed healthy. And likely the Food and Drug Administration will set national standards for acrylamide. So again, we introduced our nucleases this time into individual potato cells. So this would be similar to the embryos we heard about, or your 293 cells in your Petri dish. We take away the plant cell wall, so it's just a membrane-bound cell, and we can transform it by electroporation or polyethylene glycol transformation. Our nucleases go in. Potatoes are tetraploid, so we have 4 alleles that we need to knock out simultaneously. We treat our cells, grow them up into potato plants, and screen them. And then this gives you some idea of the frequency and efficiency of mutagenesis. 122 regenerated plants. No selection was imposed. We just took those cells and grew regenerated plants. Of those 123, 14 had mutations, and 3 of the lines had all 4 alleles knocked out. So simultaneously, we created a homozygous mutant. And we were excited when we obtained these potatoes. We only, of course, that plant only produced those triple knockout plants, or quadruple knockout plants only produced a few potatoes. But we quickly took one of them and put it in the refrigerator for a couple weeks and made potato chips. So here's our knockout potato, stored it cold and for 2 weeks at 4 degrees. Here's a wild type potato. These were chipped and fried at the exact same conditions, and you can see the caramelization that results from the production of these reducing sugars. You can actually measure the sugars, so the triple knockout or the quadruple knockout has an abundance of sucrose and very little of the reducing sugars, whereas 2 weeks in the cold, the wild type has converted most of its sucrose, broken down most of its sucrose. And then we can take those chips and measure acrylamide, and in fact, it has about 3 to 4-fold less acrylamide in these fried products. So again, through a very simple genetic manipulation, we've reduced acrylamide in the fried potatoes. And we've been able to take these potatoes out directly into the field and produce a large amount of them this year, actually, so that we can do these cold storage tests. Can our potatoes persist in cold for a longer period of time? And so in addition to the consumer benefit of less acrylamide in your fried potato, maybe hopefully we'll have also achieved a processor benefit, that is, making a potato that's easier to store. So my title was Genome Engineering Enters the Dining Room, and this is what dinner looks like at this point in time. Potatoes and soybeans, right? Not particularly the best meal. I think Scott's going to talk about some protein that can be added to this dining room table. But very soon, labs throughout the world are going to make this meal a much more appetizing and much more variety of products that are going to enter the food supply. I want to talk a little bit about now, so I've talked about sort of using gene editing to create incremental improvements to existing commodity crops. But we sort of kind of stepped back a little bit and thought about a new or a different approach of the technology, one we refer to as molecular domestication. So modern corn is derived from a wild ancestor, teosinte, this grassy, weedy plant from Central America. Most of the fruits and vegetables and grains that we use have wild relatives. And through the genomics era, over the past 10 to 15 years, we've come to appreciate the genetic changes, that which separates teosinte from cultivated maize, or wild tomato from cultivated tomato. And in fact, there are only a handful of genes that seem to be important drivers of the traits that we value. That is, changes in a handful of genes convert these wild species to their cultivated form. And so our notion is, well, can we, since we know these mutations from a variety of genomics analysis, can we take a wild tomato, for example? This is a tomato species from the Galapagos Islands. It has very small fruit, of course, but it has some traits that are of value. It's resistant to white flies. White flies carry viruses that are pretty devastating to the tomato crop. It's also salt resistant. It grows actually right along the rocks along the shore. So these are traits that could be valuable in cultivated tomato. And we know that most of the genes that have been modified, there's only a handful of genes, 6 or so genes, which when modified, and actually all of these are knockout mutations, are important for fruit shape and size, for plant architecture, the timing of the production of fruit. And so the notion is, maybe we can use genome editing to take this wild species, knock out these genes, and convert what looks like to be this wild weedy species into something that's more familiar, that's grown in the garden or in the fields. It's kind of a fanciful, perhaps naive notion, but it tests directly the role of these genes in domestication and the hypotheses that have been driven or derived from genomic analyses. And we're doing this, in fact, it's still a project that's ongoing. In this case, we're targeting all 6 genes simultaneously. So we express our guide RNAs as a single transcript in the same fashion that is done in the bacterium. And then we also express CSY4 separated by a translational skipping sequence from Cas9. So CSY4 processes all these guide RNAs, Cas9 then complexes with them and targets our genes. This is one of our primary transgenic plants. Again, as this plant's growing and developing, we're knocking out genes, and then if we take the progeny, we actually get some homozygous knockouts, so we can see a change in the plant architecture. And then in the progeny from this plant, in the next generation, we have a number of them in which we have homozygous knockouts in 4 of the 6 genes. And so, as I say, this is an ongoing experiment, but we hope that we'll be able to see if, in fact, we've achieved our desired end, if we can take these weedy wild tomato relatives and convert them into something that looks more like the cultivated form of present-day tomato. So I want to talk a little bit now about how we're employing this other pathway in plants and plant agriculture to create very precise alterations to the genome. And the challenge in plants, as in many other organisms, has been a challenge of delivery. How do we get our nuclease to make the break and our repair template in the cell simultaneously so that we can copy this information into the target locus? And we've been working on various strategies. I mean, I told you in our potato story, we took a leaf, digested away the cell wall, and made just membrane-bound protoplasts, and we were able to recover modified plants. But this approach only works for a handful of species, potato, tomato, a few others, being amenable to protoplast production and regeneration. And it's not an easy process. It takes a long time and quite a bit of tissue culture expertise. Most plants we can transform, we can get DNA into those cells. It varies from plant species to species, and it's time consuming. It's a year to year and a half from the beginning of experiment until you create the transgenic. But it's through these process of stable transformation that most genome engineering approaches take place, whether it's NEGJ-based mutagenesis or HR. And so we've been thinking of different ways to come up with different alternative delivery strategies. And one approach has been to, in animal systems, of course, viruses are very important vehicles for delivery, reagent delivery, but they really haven't been used that much in plants. And so we've been focusing on a class of plant viruses known as the Gemini viruses. They're DNA viruses, circular DNA genomes. They replicate by rolling circle replication. And they amplify to high copy number, 100 to 1,000 copies per plant cell nucleus. So they produce an abundance of viral genomes. And what we did is we sort of inactivated the virus. This is the virus, bean yellow dwarf virus. It's replicase proteins, cis-acting sequences important for replication, and then proteins that encapsulate and move the virus throughout the plant. We simply took these open reading frames and replaced them with our nuclease and our donor molecule. So we went from an infectious virus to what we refer to as a replicon, a circular plasmid, if you will, that can self-amplify and replicate in the plant genome. And so we deliver them to plant cells. This is our replicon being delivered by agrobacterium. It peels off a tDNA through the process of infecting a plant cell, delivers it to the nuclease, and then in the presence of our replicase protein, this circularizes and again amplifies to high copy number, making an abundance of your favorite nuclease to target the genome and producing an abundance of the donor to repair the targeted break. So we tested this a few years ago now in a tobacco model using a gene, a reporter gene that was integrated in the genome. If functional, this reporter has a strong promoter that expresses beta-glucuronidase, which is like LAC-Z. You can stain cells that are with GUS activity. They'll turn blue. And neomycin phosphotransferase, allowing you to select cells. So we rendered this gene inactive by removing 600 base pairs right at the fusion site, and then integrated this defective gene into the genome, and then used our replicons to make a nuclease that would break the defective reporter and provide a donor that would insert those missing 600 base pairs to restore gene function. And what we found is that the viruses, in fact, were much better at gene targeting than just sort of standard DNA delivery, on average 25-fold increase. So these are plants, independent transgenic plants that had that reporter integrated in the genome. And this is delivery by standard means. So this is gene targeting, frequency of gene targeting delivered by standard means compared to frequencies used by the replicon. And the reason the replicons worked, we were sort of able to tease this apart, was in part amplification of the reagents, as I suggested, and in particularly amplification of the donor. And these experiments sort of illustrate that. Here we have our replicon, the nuclease in red and our donor in black, versus just standard tDNA plus the replicase. So in both cases we have the replicase. And we see about a 5-fold enhancement when we amplify the nuclease in the donor. But again, it's not the nuclease that's particularly limiting. So we can, in this experiment, we again have the standard tDNA plus replicase, standard DNA delivery plus replicase. This case, we're only replicating the donor. So the nuclease is not getting amplified. And we can still see that significant enhancement. So it's mostly the donor, insufficiency of the donor that's, or the replicon's ability to make a lot of donor that allows for this increase in gene targeting. And what we observed also was actually the replicase itself has an impact, a big impact, fivefold as well. So here's standard tDNA delivery, standard tDNA plus the replicase, no replicon, no amplification of anything really, and we see this big enhancement. And what we know about the replicase is that in this cell it binds to retinoblastoma and it pushes the cells into S phase. And so we hypothesize that elevated homologous recombination occurs because HR is more proficient in S phase. And we then did experiments where we mutated the retinoblastoma binding domain and we lost this enhancement. So the replicons work through these two mechanisms, pushing the cells into a state that's conducive to homologous recombination and providing an abundance, particularly of the donor. So again, what we do now is we introduce into the plant genome a transgene that has in red our nuclease and in yellow our replicon. And the replicon is then released and amplifies in the cell and can carry out HR. So I'm going to give you an example now at the University of Minnesota that we've been working on for the past few years, where we've been trying this strategy in another important food crop, one that doesn't make it to your dining room very often, but is really the most important source of carbohydrates for the peoples of sub-Saharan Africa. This is cassava. And this program is funded by the Bill and Melinda Gates Foundation. Again, it's a gene replacement strategy, and what we're trying to achieve in cassava is improved weed control. So cassava is a weedy shrub. You take actually a piece of the stem and put it into the ground, and it'll create roots and shoots. And then over the course of about 18 months, you create a 15 to 20 foot shrub that has these tubers, roots that are packed full of carbohydrates. So, they're about the size of my arm, and you slice into them, and they look just like a potato. They're almost pure starch. But again, this is a really important food source for Africa. And weeds are a problem. In the 18 months that this takes to mature, weeds can result in 20 to 80% of reduction in yield. And the weeding that is performed is hand weeding carried out by the women and children. And so the humanitarian mission seen by the Gates Foundation is if we could have an alternative means of weed control, the women could perform other tasks and the children could get and attend school. And it's known that herbicides, chemical treatments, can achieve increases in yield comparable to four rounds of hand weeding. And so herbicide tolerance, kind of a classical product of biotech, is what we're trying to achieve in cassava. But we're not doing it through transgenesis. So again, most herbicide tolerance is created by taking a bacterial gene and inserting it into the plant genome. What we're trying to do is edit the native genes within the plant. And herbicides typically attack genes that are involved in amino acid biosynthesis, aromatic or branched chain amino acids. We do not have these enzymes. Animals do not have these enzymes. We get these amino acids from our diet. And so if you have chemical inhibitors, you really don't affect animal life, but you will starve the plants to death because they won't be able to make it and the necessary amounts of these herbicides. And over the years, we've identified in weeds that have been resistant to herbicides, mutations, amino acid substitutions, typically in the active site, that prevent binding of the chemical inhibitor. So our goal was to introduce these mutations into the cassava genome. And for the first year and a half, almost 2 years of the project, we spent a lot of time on what we refer to as transgene models. So we took, in this case, we're trying to create, this is the aromatic amino acid biosynthetic gene, EPSPS. We introduced mutations that had been found in weedy species into the cassava gene, and then introduced these modified coding sequences under the native promoter or a strong, highly expressed promoter. We introduced them into cassava to create transgenic plants prior to carrying out the gene editing. And we did this to really make sure we knew which mutations would be best, and to identify the ideal promoters for driving expression, and then also to really optimize selection methods for the gene editing events. And I want to give you some data from a few of these gene models. These are 2 amino acid substitutions in close proximity, TIPA, just 4 amino acids apart within the exon 2 of the coding sequence. And they're driven by either the native promoter or by a very strong viral promoter. Now again, these are transgenic plants that carry these constructs. And first we did a biochemical assay. So EPSPS converts shikimate into EPSP, basically the product that is then ultimately converted into the aromatic amino acids. And so the chemical glyphosate, the herbicide, inhibits this enzyme, and so you'll accumulate a lot more of the shikimate. And there's a nice colorimetric assay. So these 2 rows here show proteins extracted from wild-type plants with increasing amounts of the herbicide glyphosate. And as you inhibit this enzyme more and more, you accumulate more and more of the shikimate, and so you see the increasing pink color with more herbicide application. And then these are independent transgenic plants that carry the mutated EPSPS expressed from its native promoter. And you can see that also you'll get accumulation of these precursors. That is, the glyphosate is inhibiting this native enzyme. But if you take that same gene and overexpress it, you see very little pink accumulation. So what this means is that an abundance of this enzyme is able to convert the substrate into the product, that is the chemical inhibitor, the glyphosate, the herbicide, is ineffective in sort of inhibiting the reaction. And then, of course, we want to look at these plants out and characterize them phenotypically. I'll just jump to this slide. So here are our wild-type plants that didn't receive the herbicide, and here's a wild-type plant that did. You can see, of course, it completely killed our target plant. And these are independent transgenics expressed from the native promoter or the strong viral promoter. And so you can see you get these were all treated with the herbicide, and you can see you get very good tolerance and increased tolerance with high expression of the enzyme. And so that's just quantified here. The score is 7 is dead and 1 is unaffected. And our wild-type plant is dead after treatment after a few days. And the native promoter does less well than overexpression. So what we learned from this is we need to do 2 gene edits. We need to introduce the mutations of interest and overexpress our target locus. And that's what we've been in the process of doing. So because we have a selection, a powerful selection, we can identify, so these are cells and culture that we want to do the gene editing in, and we can identify concentrations of the herbicide that kill or impair growth. That's what's just illustrated here is growth over a period of 28 days under varying concentrations of herbicide. So we can pick a concentration that inhibits growth, and then the idea is if we edit those genes, we can grow this, they will be able to grow on those inhibiting concentrations of herbicide. And in fact, that has been the case. We've just now obtained our first gene-edited plants. So this is those cells growing in the presence of the herbicide. Occasionally we'll get a clump of tissue that grows quite effectively. And you can see those little green nodules that form. Those are individual cells that are starting to form embryos, which then germinate into full-fledged plants that can grow in the herbicide. And again, the gene models told us that we had to introduce the mutations into exon 2 and introduce a stronger promoter to drive expression. And so this plant has both. We've So we broke with the CRISPR reagent here in exon 2, and we're able to copy and replace the promoter and introduce those amino acid substitutions. And we've been able to validate that molecularly. Our plants still are not much bigger than this, but we're growing them up and multiplying them, propagating them to do greenhouse trials, and ultimately get them out into the field in much the same way that I described we did with our soybean and our potatoes. And so we're optimistic then that this will be another way that genome editing is going to enter the food supply, not in the US, but perhaps ultimately in Africa, to sort of achieve the ends of this project, the goals of this project, to reduce weeds and create cassava fields that can be weed-free basically through treatment of these herbicides, and produce more of the rooty tubers that are such an important source of carbohydrates for Africa. Yep.
Would that still be considered a transgenic GMO?
That's a really good question. We have no idea. The only rulings that have been put forth so far have been on knockout mutations. In this case, yeah, we introduced a promoter from a virus, but we're doing the same experiment wherein we're taking a strong cassava promoter. So we're adding a cassava promoter in front of a cassava gene. So it's definitely genetically modified, but how are you going to regulate it? And we would like to ask the regulatory authorities that very question and get some feedback. No one has taken a gene replacement, a product of homologous recombination, in front of a regulatory agency, in plants at least, yet. So yeah, and the other strategy would be to identify the least possible modification. That is, can we drop an enhancer in here maybe and achieve the same end? Yeah. So we're doing experiments to sort of to anticipate regulatory feedback, yeah.
How about generating a hypermorph allele?
Say it again?
Hypermorph alleles?
Hypomorph? Hypermorph, that like
more active than the native form.
Yeah. So you could think about taking that enzyme. So I talked about resistance mutations. Yeah, you could think there might be other variants that would produce aromatic amino acids more abundantly in the presence of the inhibitors. Yeah, I think it's an interesting idea. Perhaps you could beat on the gene further to get what you want so that you don't have to overexpress it. Yeah, I think it's a good idea.
Do you notice any transgene silencing in the progeny of these cassava?
Transgene silencing, that is if we introduce or duplicate sequences, for example, or?
When you make this repair, I'm especially interested in the comparison of the native promoter versus the virus promoter.
Mm-hmm.
If you would expect any silencing due to the usage of this viral promoter?
Yeah, it's a really good question, and we don't have a lot of data on that particular subject. I've seen some unpublished data actually where they went in and obtained many independent editing events, and then looked at the methylation state of DNA in the parental and then all of the edited progeny. And it actually was remarkably different and unpredictable. Some were completely like the parent methylation state. Some had completely or missing the methylation. So I think epigenetic consequences of editing is still kind of a very open area. And yeah, a lot of times, they're sensitive. Epigenetic states change in response to sort of copy number. Are the replicons I described go in and amplify to very high copy number? Does that have an epigenetic consequence for the locus? These are all questions we need to think about, yeah. Okay, in the last few minutes, I'm just going to tell another sort of short story in which we're sort of harnessing the CRISPR-Cas system as an antiviral agent, really. I mean, and that's its natural purpose. So bacterial cell, you have your CRISPR array that makes the guides, that complex with Casps like Cas9, and then this archive of information is information representing past DNA invaders, other viruses or plasmids, so that if you're infected by another agent like another virus, you have a guide RNA that'll recognize it, cleave it, and thwart infection. So we said, well, plant viruses, cause a huge loss in crop productivity every year. What if we just insert Cas9 into the genome along with some guide RNAs that target plant viruses? Maybe we can take this bacterial immune system and use it in plants. And so, I talked about bean yellow dwarf virus and how we harnessed it as a replicon. Well, in this case, we wanted to see if we could inhibit infection of the native virus. So what we did is we actually, in a test system, is we replaced one of the proteins with GFP so that we could measure replication of the replicon, and then we targeted a number of guide RNAs to conserve sites, either in the coding sequences or the cis-acting sequences that are important for this virus to move in the plants. And we did some initial tests just to see if it was worth pursuing this approach. So here we have 2 strains of Agrobacterium. Agrobacterium will deliver our DNA to the plant cell. And one Agrobacterium strain, we have our guide RNAs and our Cas9. In another bacterial strain, we have our replicon, which has our GFP. So the idea is if you infiltrate a leaf, you can then look after a few days to see the amount of GFP fluorescence. And if you've targeted and knocked down GFP, you'll be able to quantify the amount of that knockdown based on the amount of fluorescence. So this is GFP intensity targeting these guide RNAs that target different parts of the viral genome. So this is a guide that doesn't target the genome, and so we normalized it to 1. And you can see that some of the guides very effectively inhibit GFP production, and particularly if you use multiple guide RNAs. And then what we also did is we took these cells, we took the punch and we made DNA, a leaf punch, and made DNA from it. And then, since it's a circular genome, we just transformed E. coli and counted the number of colony-forming units. And again, the amount of circular DNA in those leaves correlated really well with GFP intensity, suggesting that the loss of GFP intensity was due to knocking down replication. And then that gave us the motivation to then make stable transgenic plants that had Cas9 and guide RNA targeting the DNA viruses. And we then infected those plants and then measured the amount of viral load. And again, we saw less virus in the plants that were expressing the guide RNAs. And the real proof in the pudding was when we actually, when you could actually see the phenotype. So this is a plant that's only expressing Cas9. It's being infected by the virus, 21, 28, 35 days. You start to see the yellow wrinkly leaf phenotype. This is in contrast to a plant that's expressing Cas9 and a single guide RNA that targets the virus. And you can see that you have much reduced symptoms of viral infection. And so, a lot of people are interested in using this to thwart DNA viruses. You're probably aware now that there have been a number of, or there have been Cas systems described that target RNA. Perhaps those could be used to target RNA viruses, which are even more important pathogens of plants. So, I think, we can learn from what the bacteria did, perhaps, and harness these systems to help combat plant disease. So I think I'll just close there and just a few very big high-level points, take-home messages. At the beginning of my talk, I talked about how we're the first stage of genome engineering in plant agriculture, which is all about the knockout mutations, the soybean, the potato. And government authorities have weighed in on those and said, at least in the US, Argentina, Israel now, that such mutations, knockout mutations, plants with knockout mutations, are not regulated and can get out into the field and eventually into the food supply. We still have a ways to go to make gene replacement efficient in plants. I talked about our replicons, which are helpful, and some of the experiments that we've undertaken in cassava to make some of the first products of gene replacement, also those that will have an impact, hopefully, on agriculture and the food supply. The potato and soybean project were carried out by the scientific staff at Calyx. This is my lab at the University of Minnesota, who did really the rest of the work that I talked about today. So thank you. I'd be happy to answer any questions.